Yeah, coming back to the discussion on the cell material interaction, let me recapitulate what I have covered in the last module. So, cell adhesion, it involves the first is the involves the adsorption of the protein molecules, and the cell adhesion proteins can bind to other molecules or ligands, the extracellular matrix, and this protein adsorption takes place at the beginning at the within first few minutes of the cell culture experiments. Now, after proteins absorbed with specific conformation of the material, that specific conformation I have emphasized also in the last module, that is necessary because afterwards protein has to interact with the cell surface receptor proteins, and cell surface receptor proteins interact also with these adsorbed proteins. Now, this protein adsorption as I mentioned in the last module, that protein adsorption, proteins are of different types which are which get adsorbed on the biomaterial substrate, and this protein adsorption typically follows Langmuir kind of isotherm. If you plot the protein absorption as a function of the initial protein concentration, you will get this kind of curve which reaches the steady state behavior. So, this is on this Langmuir isotherm behavior, and similar protein adsorption kinetics you can also explain in terms of kinetic dependence of the absorption if you plot the protein adsorption as a function of time. So, initially the monolayer proteins get absorbed, and then after that protein molecule. So, here it goes. So, this is the spreading particle model, what you see that density of the proteins, and then as a function of time of adsorption, if you plot, then it also reaches a steady state behavior. So, mathematically Langmuir isotherm can be explained by this particular equation, where Cs is the adsorbed protein density, Cb is your bulk protein density, K is your adsorption coefficient, M is the molecular weight of the specific protein of that you are concerned, and capital A is your surface area of the biomaterial substrate to which protein is exposed to. So, this is not a very linear type of equation. As you can see, Cs depends on the factor that Kcb by 1 by divided by 1 plus Kcb. This is the second expression which is also equally important in describing this Langmuir isotherms, that is d rho by dt, that is the change in the protein adsorption density with respect to the time of incubation, is equal to two terms. The first term is related to the adsorption, the second term related to the desorption. So, absorption terms, as you can see here, that Ka term, that is the adsorption coefficient, capital C is your bulk concentration of protein, which I have explained in the last equation as Cb term. So, here you can see this is the bulk concentration Cb, the same thing I am mentioned here C, or I can explain as that Cb term into 1 minus rho upon rho monolayer. So, rho monolayer is the monolayer protein concentration, rho is the concentration of the protein at any time t, Kd is the protein desorption coefficient, and rho is again absorbed protein density. So, this expression is valid only when your initial seeding concentration of the protein is kept constant, and this expression is valid at any given time t when t is constant and protein adsorption follows this Langmuir isotherm. Okay, this slide is essentially a recapitulation of what I have mentioned to you in the last module, the cell growth and proliferation. So, this is a typical experiment that has been shown in it without any biomaterial substrate on a cell culture tissue culture plastic, which is used as a reference or control sample in all the cell culture experiments. Now, what you see here, that initially cells start proliferating, then it forms a confluent monolayer, and when this press medium is pumped across the cells, and then flow of the medium also simulates cell proliferation. So, if you give some media flow, So, it is not a static culture, it is little bit more dynamic culture. So, if you give some flow, that means you are essentially giving some shear to the suspension culture, and therefore, cells numbers also would increase. So, tissue can grow by increasing the number of cells or size of the cells or intracellular substance. An increase in the cell number with increase in the culture duration is by far the most important criteria to quantify or to establish cell growth, which I have sufficiently explained in the last module. So, observation of cell morphological observation or quantification of the cells adhered to a biomaterial substrate needs to be done from a microscopic study. Here you see this is a 1 day, after 1 day this is your number of cells, and this is your 7 days the total number of cells, it is spreading all along the surface. So, this is the one of the very slowest growing cells, that is the L99 mouse fibroblast cells. So, if you now plot, if you have some observations at day 3 or day 5, and if you now plot this cell number, certainly you can see that this cell growth is kind of shows the very linear pattern. Now, one of the things in the last module I have mentioned that once a biological cell adheres to a material substrate, it sends soluble signaling proteins to neighboring cells, and cells after receiving the soluble signaling protein signaling molecules, they decide whether to go towards the biomaterial substrate or not, or they decide whether to adhere to a biomaterial substrate or not. Once this cell once these cells will adsorb on a biomaterial substrate, then there is an opportunity for the two neighboring cells also to interact with each other. In other words, these two neighboring cells, their cell surface proteins which can be exposed to each other, they also get an opportunity to interact with each other. So, therefore, that cell to cell interaction is being discussed in this slide. Now, what you see here, this is called homeophilic binding. Homeophilic binding means that so this is the cell surface receptors from cell A and cell B, which are of the same cell type, and this is the similar kind of cell surface receptors they come. So, they bind very easily with each other. Now, heterophilic binding means that there is two different type of cell surface receptors which is coming from two different cells, this A, C and D, let us say, and then this is called heterophilic binding. Now, if this direct binding of two neighboring cell surface receptors is not possible, and it has to be mediated by a extracellular linker molecule, that is also possible. So, this is called type 3. So, this type 3 mechanism is by far is less commonly observed mechanism which can explain the cell interaction. The other things which is important for you to realize that once a cell adheres on a biomaterial substrate, cells also secrete some of the proteins molecules and some of the biological macromolecules which gets out of the cell to the exocytosis process to form extracellular matrix, which as I have explained in the last module as well. Once this extracellular matrix they form, which essentially composed of the elastin fibers as well as this fibrous collagen protein. So, this extracellular matrix has been shown here the way I am drawing here, or I am trying to sketch here. Now, once this extracellular matrix forms, the orientation of the collagen fibers and the orientation of the actin filaments, as you can see, they are oriented almost parallel to each other. However, in the neighborhood of this cell number 1, if there are 4 other cells are there, 2, 3, 4, 5, if you notice their actin filaments or their cytoskeleton fibers, they are oriented quite differently with respect to the ECM around cell number 1. What it means that there is a clear mismatch in terms of their orientation between the intracellular cytoskeletons and extracellular matrix collagen here. Now, if the more and more extracellular matrix is synthesized around the cells, and therefore, in order to provide sufficient support to these particular cells. So, you remember that one of the functions of the extracellular matrix to provide mechanical support or structural support to the cells it contain. So, therefore, this interaction or this mechanical support will be much more effective if and only if that collagen fibers in the extracellular matrix are oriented almost in the parallel to that of the cell in the cell number 1. So, in the next slide, as you can see that as the extracellular matrix progresses, formation progresses, then this cell number 2 here and cell number 3, their cytoskeleton reorganization take place, and as I said that this is one of the way that cell kind of adapts to itself by cell shape changes, and that is what exactly happens. If you see the shell shape here in that marked as 2 and 3, and if you see the shell shape here marked as 2 and 3, you would be able to realize a distinct difference in terms of the cell shape and cytoskeleton orientation. And this cytoskeleton reorganization has been helpful now for ECM to provide more mechanical or structural support to cell number 2 and cell number 3. However, two other cells which are also shown here, cell number 4 and cell number 5, their cytoskeleton is still not oriented with respect to the extracellular matrix. Now, what happens therefore in the next stage? In the next phase, as you can see here, that cell number 4 and cell number 5, their cytoskeleton is also oriented now with respect to the extracellular matrix here. And now, cell number 1, 2, 3, 4, 5, all 5 cells are oriented perfectly with respect to the extracellular matrix, and that is what is mostly biologically desired, or this is the scenario that would help extracellular matrix now to provide sufficient structural support to all 5 cells together. Now, one of the techniques that I have mentioned to you earlier is that fluorescence microscope. This is one of the typical images of the fluorescence microscope, cell images which you can grab using the fluorescence microscope. Now, what you see here, the cytoskeleton organization within the cells, you can see in the green stained fibers, this is your actin filamentous fibers. You also see the blue colored region, this is your DAPI, DAPI stained nucleus, and nucleus means you have a DNA. So, DNA intercalates with the DAPI stain, and therefore, it keeps this blue colored. You also see somewhere this red colored dotted line, dotted points. So, this is like small organelles, and these organelles are essentially mitochondria. This mitochondria can be stained with microtracker, and with this staining that you can see here very clearly at least 3 organelles, 3 structural part. One is the cytoskeleton actin filament, one is nucleus, and one is mitochondria. Okay. Now, in reference to the significant discussion that I had in the last module, that once a cell adheres on a material substrate through these focal adhesion complexes. So, focal adhesion complexes, and these focal adhesion complexes, after that what will happen, that whether there are couple of opportunities or there are couple of options that the cell would have. One is that whether the cell would replicate. Replicate means it will self propagate, like this cell would give rise to another daughter cell. So, 2 daughter cells, or cell would differentiate to another cell type, that is a different cell type. And in traditional biology, if you want to show that a mother cell or a parent cell is differentiated to another cell type, you must show nucleus with different color or different shape or different shaded nucleus, just to distinguish that this nucleus is different between these two cell nucleus are different one is that undifferentiated cell and another one is differentiated cell. Number 3, if you remove all the cell signaling processes in the neighborhood of this isolated cell, then cell can also activate its own suicidal mechanism to undergo cell death. I will tell you later that cell death normally takes place by 2 processes. One is called cell apoptosis, that is called programmed cell death. Another one is called cell necrosis, that is called accidental cell death. So, which is essentially due to the physical damage or to the tissue or cells. Number 4 is cell motion or cell migration or cell motility. So, this motility, cell motility, cell migration, these are all synonymous terms. Okay. So, cell migration means like a human being walks on the ground or the road, similarly the cell also can crawl on this particular biomaterial substrate. So, this kind of transport or the going from one place to another on another non-biological synthetic surface, this process is known as the cell migration. And 5th one is cell adhesion that I have mentioned earlier. Now, all these processes, what I say, all these processes, that cell replication, cell differentiation, cell death, they are grouped under one particular name called cell fate processes. So, cell fate essentially means that when a cell is being cultured on a biomaterial substrate, what would be its fate? Means whether the cell would grow, whether the cell would differentiate, whether cell would not be able to survive, or cell would adhere to the material substrate, or cell would migrate, or cell would become mobile on the biomaterial substrate. Remember, this cell migration or cell motility is kinetically extremely slow process. I will give you some numbers in the later on, and then you will realize that typically cell migration takes place with a speed of several microns per hour. That means, in a single hour a cell can move ahead by the process of migration only few microns. Few microns means 10 to the power minus 6 meter. So, then you can realize that how slow is the cell migration process. Now, all these cell fate processes are essentially the result of a coordinated cell signaling process. So, therefore, one has to also define what is cell signaling process. So, cell signaling process is nothing but a part of the cell communication, and the cell communication is essential to coordinate all the cellular activities, and it occurs in three principal ways. One is the soluble signal, second one is that insoluble signal, third one is a direct cell to cell contact. I will explain to you one by one, but first you see that what is soluble signals. So, soluble signals means like cytokines, like these are like growth factors. So, therefore, one has to explain what is growth factors also. So, growth factors are extremely small protein molecules which can stimulate certain cell fate processes like proliferation and so on. So, these small protein molecules means their molecular weight can be few kilodalton. So, chemokines is one of the growth factors that induce cell migration. Cytokines is one of the growth factors that classically cause proliferation and differentiation. But there are 3 type of growth factors also important soluble proteins. Autocrine, auto means similar type of cells, paracrine means cell signal to neighboring signals, and endocrine means cell secretes growth factors which is transported through bloodstream and carried into a target cell. So, essentially cell signaling means you have a source cell and you have a target cell. So, source cell and target cell here, these signaling molecules. So, here signaling molecules can be transported to the from source cell to target cell, and as a result this cell signaling process, the cell communication takes place, and this cell signaling processes leads to the several cell signaling processes enable several set phase processes to take place. So, one of the cell signaling processes is through the tight junctions or gap junctions. These tight junctions or gap junctions are commonly seen in case of epithelial cells. Epithelial means that is the skin tissue cells. So, and then also this is any closely spaced cells, there the cell signaling can take place very easily, as you can see from here to here the cell signaling process take place. So, this is two neighboring cells, signaling cell and target cell, it is in the physical proximity. So, this is the example of direct cell to cell contact. What is the other things that I was trying to tell you is that from source cell to target cell, this process can take place by diffusion, or if there is any some blood vessels are there. So, these signaling molecules can be transported through the blood vessels and can get carried to the target cell also. So, this is the kind of third one, that is the endocrine signaling process, which I just explained. And if the signaling process goes by diffusion from one source cell to another target cell, then it is called paracrine signaling process. I mentioned about the growth factors. Here it is clearly mentioned, it is a small proteins of the order of size 15 to 20 kilodalton in size, and 1 dalton is equivalent to one weight of 1 hydrogen atom. So, the functions of the growth factors, that it can stimulate the cell growth, that is increasing in cell numbers and so on. There is other growth factors are there, various type of growth factors, which you may not remember all of them, but a few of them are being highlighted here. One is fibroblast growth factors. So, that is responsible for skeletal and nervous system development. You have insulin like growth factors, and another one you have transforming growth factor beta. So, that regulates cell growth and differentiation of many cell types. So, these three growth factors, perhaps it is useful to remember in the context of bone tissue engineering applications. So, this is what I was mentioning. Paracrine means it goes to diffusion, this is your source cell or signaling cell, and this is your target cell. So, there are 3 target cells here, this is target cell number 2, this is target cell number 3, and from the source cell the small signaling molecules is kind of transported through the target cell slowly. And what you notice here, the biological interaction is actually takes place not directly, but through cell surface receptor proteins. So, the signaling molecules they go, and then they will get they will immediately being tagged to the cell surface receptors. So, here again cell surface receptor is kind of being tagged to the signaling molecules, and as a result there is some downstream signaling mechanisms get activated in these target cells, and that downstream signaling mechanisms, once it is activated, they can help in the protein synthesis inside this target cell. They can also, if they are transported significantly and getting into the nucleus of the cell, then they can alter the gene expression of the target cell also, therefore, enables the differentiation process to take place. So, I have explained these things in a very nutshell or very brief, that how signaling molecules, when they are being transported, they can activate the protein synthesis, they can activate the cell differentiation process and so on. So, this is another type of signaling process, that is called synaptic signaling. So, synaptic signaling essentially means you have a neuron. So, it is a very long characteristic structure. So, you have a neuron, you have an axon here, this is a cell body and nucleus, and you have a synapse, and the synapse is there, and you have a target cell T is there. So, here again signaling molecules or neurotransmitters, once it is transported through towards the target cell, then the cell surface receptors of the target cell will kind of be hooked to the signaling molecules, and as a result certain downstream signaling processes gets activated, and that will influence the way this target cell would otherwise function. Paracrine signaling process, the endocrine signaling process I have mentioned. So, this is your source cell and this is your target cell. There is two target cell that has been shown here. So, these signaling molecules being transported to the bloodstream, to the blood vessels, and they can transport it towards the target cell. They get tagged to the cell surface receptors in both the target cell, and thereafter they will influence the cell functionality of the target cell. And this is the gap junctions here, like how these signaling molecules are being transported through the channels which is present at the cell membrane level itself in both the cell membranes, and they are so physically or tightly spaced, these two cells. So, you cannot even distinguish that which one is source cell and which one is target cell. For general understanding, if you consider the left hand one is the source cell, right hand one target cell, then what happens? We are trying to understand that once the signaling molecules get transported through this porous channel, what would be the kinetics of this cell signaling molecules, and how many number of cell signaling molecules can pass through this kind of gap junction. That is what I will explain to you maybe in the next module.